Hello biology students, and welcome to the first video in our evolution unit. So in this video, we're going to be talking about something a little bit different than in the rest of the unit, and that is the history of life. So in other words, how life was first formed on this planet. I'm going to take you back a little bit first. So I want you to imagine the Earth 4.6 billion years ago. So at this point, the Earth was very inhospitable to life. In other words, we didn't have conditions that were ideal for living things. So to kind of paint the picture a little bit, there were a lot of volcanoes. The Earth's core was very active. We had lots of magma coming up, lots of lightning. At this point, there was no oxygen on the planet, and because there was no oxygen, we did not have the ozone, because the ozone layer is made of oxygen. It's O3. So therefore, we had a ton of UV radiation hitting our planet and very, very high temperatures. So you can kind of think, this is not ideal for life because a lot, not all, but a lot of life requires oxygen, and UV radiation will lead to massive rates of mutation. We have to have a very stable temperature to be able to survive. So life really wasn't going to exist in these conditions. Now, that was 4.6 billion years ago. So about 700 million years later, so 3.9 billion years ago, is when scientists think life first developed from non-living materials. So we look at a little timeline here. You can see, we think somewhere between this 4 billion and 3.5 billion is where the origin of life first happened. It's hard to tell because we use fossil records to figure out a lot of these things, and the first living organisms were so small and so simple they had nothing to fossilize. So this is when we're estimating based on what we looked at in the rest of the timeline. We do know, mostly from the fossil record, that we saw the first eukaryotes around 1.9 billion years ago. We see the first prokaryotic fossils around 3.5 billion years ago. Multicellular organisms around 1.5, and then animals didn't come until about 0.6 billion years, about 600 million years ago. Okay, so how did life originate? So there were 4 main steps for this process to happen. First, we built monomers from inorganic substances. So we took inorganic things that do not support life and combined them to make our monomers, so to make our monosaccharides and our fatty acids and our nucleotides. Stage 2, these monomers were formed into polymers. Stage 3 is these polymers were packaged into what we call protobionts, which are like little cells that aren't actually cells. They just kind of look like cells. And then finally, these protobionts gain the ability to self-replicate, because we know the ability to replicate themselves is a requirement for living things. Okay, so let's talk about step 1 first. So remember, step 1 is when we take small inorganic molecules, so molecules that do not support life, and combine them in ways that creates our small organic molecules, which remember are our monomers. Okay, so what we think happens is that we know that certain inorganic chemicals like carbon dioxide, methane, ammonia, hydrogen gas, hydrogen sulfide, and water, they were around because we know they're released from volcanic eruptions, and we know there were volcanoes at this time for a fact. And the hypothesis is that these things were combined using energy either by lightning or by UV radiation, because we know we need some energy to combine things to create larger molecules like monomers. And normally, nowadays, that comes from an enzyme lowering the activation energy, but we didn't have enzymes back in this time because there were no proteins. We also hypothesized that as Earth cooled, all this water vapor that was being released from the volcanoes condensed and became the oceans. So I don't know about you, but to me, this seems a little far-fetched if I just heard just this information. But it turns out there have been experiments that have proven that not only is this possible, but it actually could happen fairly rapidly, rapid being a relative term here. So we have a scientist by the name of Stanley Miller, and he basically showed that if you had all those inorganic compounds, the methane, the ammonia, the carbon dioxide, the hydrogen gas that we talked about in the previous slide, and you put them under the right conditions, we can create organic materials without the use of enzymes. So let me explain his experiment. basically he had this little apparatus here that he had some water, because we knew water existed, and he would heat it up so the water would vaporize, turn to water vapor. And then he had a chamber here that was full of all those gases that I mentioned, so things like methane and ammonia and hydrogen gas. And because it's far from the heat source, the water would then condense and go back into liquid water. So that's kind of like our water cycle. And what he did in the chamber over here is he added electricity. Basically, he simulated lightning, because we knew there was lightning at this point. And, as the water vapor goes around, interacts with these chemicals and the electricity and condenses, some of the water is put into a little beaker that's taken for analysis. So he let this run for quite a while, weeks, months. And basically, when he analyzed this water, he saw that there were amino acids, there were sugars, there were lipids, there were nucleotides, there was ATP. All of these things had formed by these molecules spontaneously combining. And this experiment has been replicated many, many, many times, and we see the same results every time, is that we're making monomers from these simple inorganic compounds. So pretty cool. By the way, he also set up a control that had no electricity, and it turns out it does not work without electricity. So again, this showed that really you needed those conditions that we talked about in the first slide to allow this to happen. Okay, so that's step one. So now we have some monomers, but great, monomers do not mean we have living things. We just have a bunch of glucose and a bunch of fatty acids and a bunch of amino acids. So kind of the next step is these basic monomers were turned into polymers. Now, if you think back to earlier in the year, we know this can happen via dehydration synthesis, but dehydration synthesis uses an enzyme, and we don't have an enzyme because enzymes are made out of protein, and proteins are a polymer. So really the question is, how could this happen? And it turns out that, remember, enzymes just speed up chemical reactions. Chemical reactions can happen without enzymes. They just happen a lot more infrequently and a lot more slowly. So if I had two glucose monomers, oh, that's a terrible drawing, that's okay. And these combined in just the right way, and they hit one another in space in just the right way, that they can react, they could form a disaccharide. It's just not very likely. So if left for enough time, monomers will polymerize. They will turn into polymers, and we know this. We've done experiments to show this. And we hypothesize that because the Earth was so hot and because the oceans were fairly shallow at this point, that if you have a very hot environment, like we talked about with high temperatures, water is going to be constantly vaporizing. So we're going to have very shallow puddles and lakes, which means we have less water, which means we have a more concentrated solution of monomers. And if the solution is more concentrated, they're more likely to hit one another in the correct orientation and polymerize. Okay, so that was step two. So now we have a bunch of polymers. Fantastic. So we have some proteins, we maybe have some RNA, have some polysaccharides, and we know we have some lipids. And it turns out if you have lipids and you just put them together, they will spontaneously form a membrane because the nonpolar and polar pieces will orient in the correct direction to get all the nonpolar bits together and all the polar bits together. And you can kind of imagine if I have my little phospholipids that have formed in the previous step, and if they form a little membrane like this, they'll spontaneously form little packages. This is a terrible drawing. But you can see I'm getting a circular membrane going on here. And if they just by chance catch some other molecules inside of them, so maybe I have my little lipid circular membrane thing going on here, and maybe in the inside of this circle, maybe I catch something. Maybe it catches a polysaccharide or it catches an RNA molecule. So now I have different things on the inside and my outside of my cell. And this is you can kind of looking at it, it's starting to look like a cell. It's not a cell yet, but it's starting to look like one. So this little container here, this little lipid package, is called a protobiont. Okay, and we think that these protobionts, if they captured some RNA, well, now they have some genetic material. And if they capture some protein, well, maybe they could then evolve to become the first prokaryotic cells. Pretty cool. Okay, and then finally, the last step 4, so we have these little protobionts, but they have to be able to replicate themselves. And it turns out, it's pretty cool, there are these things called ribozymes that are just pieces of RNA, and we know RNA could exist at this point because the Miller experiment showed that it could. Turns out some very short RNA sequences that wouldn't be hard to make from scratch can actually replicate themselves. What happens is the RNA sequence will spontaneously hydrogen bond with the complementary sequence, and then I have the opposite sequence of what I originally had. And then if this complementary bonds with its complement, you can see how eventually I will get a replicated piece of the original. And this happens. There are little RNA, what we call RNA enzymes, ribosomes, that can do this. So scientists are very confident that the first genes in these little protobionts were actually RNA molecules that just by chance were able to replicate themselves. You can see we're starting to get genetic material that's able to be replicated. Okay? And you can kind of imagine, well then maybe eventually just by chance I formed a tRNA molecule that was able to carry amino acids and started to bring in pieces of protein. So therefore the RNA could act as a template to build a polypeptide, a simple one of course, and then maybe just by chance it created a polypeptide that could help with replication. And over the course of billions of years, remember we're talking a long, long time for this to happen, we eventually get proteins that are functional and can do jobs for us. And you can kind of see how very, very simple prokaryotes might have evolved. Okay. So that is how we think the first cells came to be. And this is a pretty widely accepted theory. So now we have these simple prokaryotic cells. We have to kind of think, well, how did more complex eukaryotic cells and multicellular organisms come to evolve from these prokaryotes? So I want you to take a second, pause the video, and try to think about the three processes that are listed here. So photosynthesis, fermentation, which is anaerobic respiration, and aerobic respiration, or cellular respiration. I want you to think about which one's involved in what order and why. So go ahead and pause it. Okay, so now let's talk about what the correct answer is. So fermentation is a simple way, it's a simple way to take glucose and make energy. So because this is so simple, this probably evolved first. Aerobic respiration is much more complicated, but it requires oxygen. Remember, we don't have any oxygen at this point, and photosynthesis creates oxygen. So photosynthesis probably came second, and that probably created the oxygen that allowed an aerobic respiration to occur. Okay, so let's talk a little bit more about this now. So I want to set the scene again. So remember, at this point, so we have some basic prokaryotes existing, but we still have little to no oxygen gas. We know from the fossil record that basically the cells that existed were prokaryotic, or at least they were the basic prokaryotic cells. And we know the environment at this point is full of organic molecules because all those inorganic molecules have been combining to make monomers and polymers, and they're all floating around in the ocean that exists. So our first cells were anaerobic, that means they did not use oxygen. They were heterotrophic, which means they got their energy from all this, by eating all this food that's been floating around, and we know they were prokaryotic. Okay, so basically what happens now, these heterotrophs that get their energy from eating all these molecules, they proliferate and divide until most of the organic molecules get consumed. So now, any organism that has a mutation that makes them get energy from the sun or from chemicals would have an advantage because they could make their own food. So now we get the evolution of chemosynthesis, which is producing glucose by using the energy of inorganic substances like sulfur. And we get the evolution of photosynthesis, little tiny cells, bacteria that can get energy from the sun to make glucose. Okay, and we know one of the byproducts of this process is oxygen. So oxygen is released into the atmosphere. Now this creates an even bigger problem. We have evidence for this happening 3 billion years ago. We have photosynthetic bacteria at this point. However, oxygen, it turns out, is pretty bad for you. It can damage enzymes. It turns out our bodies have a ton of ways to deal with the toxic effects of oxygen because as helpful as it is, it's also very harmful. So because oxygen is so damaging, some organisms evolved the ability to bind oxygen to other compounds so that way it can't hurt their enzymes, just like we bind oxygen to hemoglobin in our blood so that way it can't hurt other things when it's being transported. And this is the first step of aerobic respiration. So you can kind of see from photosynthesis, we got the evolution of aerobic respiration. It took about 1 billion years for oxygen to develop to what the levels we have today, but eventually, once it got to present-day levels, it reached the upper atmosphere, formed O3, which is the main component of the ozone layer, which helped to protect our organisms from UV damage that could damage the RNA and DNA that they had established. Okay. So finally, the last thing we're going to discuss is how we went from prokaryotes who can do photosynthesis and prokaryotes who can do cellular respiration to getting eukaryotes. So we think, pretty simply, the nucleus just came from, at one point, the plasma membrane folding inward and creating an extra little compartment inside the cell. But how we got cells that had organelles is much more complicated. And this we explain using the theory of endosymbiosis. Basically, the idea is that a large anaerobic eukaryotic cell, so a large cell that is anaerobic, so it can't deal with oxygen, but it's eukaryotic, so it has a nucleus, engulfed or ate a small aerobic prokaryote. So it took in a prokaryote that could deal with oxygen, that could do cellular respiration. So this would be mutually beneficial because the little aerobic prokaryote would live inside the big anaerobe and have a beneficial environment, and the bigger one will be taking energy from the smaller one because the smaller one can do cellular respiration and make ATP. And we think eventually, over millions and millions of years, this small prokaryote eventually evolved into the mitochondria. Then later on, the same cell, so my big anaerobic cell that now has a small aerobic prokaryote living inside of it as a mitochondria, eventually engulfed a prokaryote that could do photosynthesis, and that eventually evolved into the chloroplast. So let's look at a picture here. So here I have my little cell that is prokaryotic and it can't do cellular respiration or photosynthesis. Eventually, part of its membrane folds in and forms the nucleus over several million years. And it gets its energy by eating other things. And eventually, at one point, it goes to eat a little bacteria that can do cellular respiration, and for some reason, it doesn't break it down. And this little bacteria keeps doing cellular respiration inside the cell and makes ATP, and this cell just takes the ATP and it gives it a little home. So it's a little symbiotic relationship. And, they continue to divide together, so whenever the big one divides, the little one divides, and over millions of years they live together. And then eventually, the ancestor of this cell, so several million years later, this cell engulfs and goes to eat a little cell that does photosynthesis, and again, for some reason, it doesn't break it down, and that eventually becomes the chloroplast. Okay, and there's a ton of evidence for endosymbiosis. This is a very, very, very well-accepted theory at this point. Turns out replication of our mitochondria and plants' chloroplasts to this day replicate independently of the cell cycle. So this implies they were once autonomous, free-living things that they still kind of act a little bit autonomously. Very strangely, these two organelles have their own DNA. They're the only organelles that have DNA in them other than the nucleus, which implies that they came with their own DNA because they were once independent material. Also, the DNA that's found in these two organelles is circular, which is only prokaryotes have circular DNA. So again, it implies they were once prokaryotes. They also have two membranes, because you can kind of picture if I have my big cell and my little mitochondria, if my big cell goes to eat the little mitochondria, it's going to end up with its original membrane, and then this membrane has a second membrane. Both these organelles have two membranes, which again implies they were engulfed. So that's it for the history of life, and I'll see you in video 2.